We've seen a variety of genes that have an effect on the organism, the phenotype of the organism, and we've also kind of entered in with the genes that have big effects, and kind of underlying that, we got into non-Mendelian and some genes that might have a little bit lesser effect. Of course, with all things in biology, there's a spectrum of full effects, big effects, all the way down to hardly any effect. And a lot of the genes we study for, the ones we've talked about in lab and the Mendel experiment, pea plant genes, they're just kind of quirky little differences in color or shape or something like that. But under that all is the expectation of like function and organism health. So I want to kind of get a perspective on that here with how important an individual gene is and why, and how the individual essential genes, the ones really important to life, relate to the abundance of the maybe a little bit less essential genes. So topic of essential genes and also non-essential genes. So I think sometimes maybe this term essential gets a little muddled. So essential is related to essence or being, really. So when we talk about essential genes, these are genes that have a function that is required for the organism to be alive. So whether that's a single-celled organism all the way up to a complex multicellular organism, there can be some of the genes that you just can't do away with. You can't mutate them. Some of them you can hardly mutate at all. Other ones you wouldn't be able to remove the gene, but it might be able to mutate a little bit. So just hard to kind of put that all into one context here, but maybe give you a little range of expectations here. So obviously we have a lot of interest in studying humans and human genetics. So this is a little bit of context for the human genome. 3.1 billion base pairs. Those are the nucleotides that make up our chromosomes. And about, this number ranges depending on who you ask and when you ask, about 25,000 genes. So this includes genes we know their functions and genes that have suspected functions, all the way down to pieces of DNA that look like genes, but we don't know anything about their function at this point. And this headline over here is kind of an interesting one. This is just from this year, 2021, where it's been about 20 years after they declared the full sequence of the human genome and this Human Genome Project. But basically, as part of declaring victory on that Human Genome Project, they left out a few harder to sequence, harder to really nail down pieces of DNA. So those are finally determined now, and I don't know if those contain genes, but certainly nice to have that even more complete now than it was before. So humans, of course, are multicellular eukaryotes, and we have a variety of genetic tools, I guess, that are distinct for our species and some that are distinct for multicellular animals, mammals, and so on. And one way to look at essential genes, it's a little bit easier to look for them in organisms that can be more easily manipulated, that are a little more expendable. And for humans, we have this cell culture ability where you can take cells out of a human and grow them under careful lab conditions, where you sort of simulate the internal environment of a body, and the individual cells, they get what they need. They have oxygen and nutrients and temperature conditions that are good for them, proper chemicals they're used to be surrounded by, and you can grow them up. So you can grow cells from different parts of the body. Some of the first human cells to be cultured were human tumor cells. So some of these cells that are a little more aggressive to grow in the first place can be grown on their own. And in this context, obviously, whether you have a tumor cell or a functioning normal human body cell, it has to have the tools that it needs to be alive. So there's kind of a minimum of what it takes to be alive. And under this kind of condition, with cells that are carefully taken care of in a petri dish, you can challenge them a little bit by removing genetic information and seeing what you can change and kill the cell versus what you could change and still have a cell that's viable and alive. So this kind of gets at the bare minimum more on a cellular level. So it doesn't have to do with the whole organism function. Obviously, we have organs and many different cell types that have to work together to keep an organism alive. But even just a human cell has, as we see here, about 2,000 genes that are essential for cell survival. This is another perspective. I really enjoy this study from not too long ago, where they took a bacterial cell starting point, and same thing as I was describing before, this is a cell that normally lives on its own. And bacteria, by comparison to eukaryotes, have smaller genomes. They've got less DNA to work with to begin with. They're kind of a streamlined organism. And what they did in this study, same thing as I mentioned before, they deleted a gene, deleted gene regions, and they took parts out, and they said, is this still alive? Okay, we'll take something out. Is it still alive? Take it out, and so on. So this is this kind of circular process of taking apart bits of the genome, and eventually they stripped it down like the least genetic information you could have and still have a cell alive. And they got it down to what's being shown over here, this 473 genes. And starting with a rather simple cell to begin with, you could probably take this as about the minimum that it would take for a cell to be alive. So a little less than 500 genes, 531,000 base pairs, pretty darn small. So the reality of a lot of genomes is that they're much bigger than this, and they have more DNA than they need strictly to be alive. We'll kind of talk about that in a little bit. One of the perspectives as far as what it takes to be strictly alive is the fact that these experiments are done in a laboratory. And in a laboratory, even if you take a single-celled organism like a bacterial cell or what's shown here, yeast cells, if you put them into careful lab conditions, they have all that they need, and they don't have challenges from the outside. So living, as we know, for organisms, living is challenging, and without having to face those challenges, the individual cells don't need certain tools that they would use otherwise to collect resources, to defend themselves, to communicate with other cells, and so on. So there's sort of the minimum, and I call them here pampered lab cells under perfect, ideal conditions, right? So if everyone's providing all that you need, you get a little soft as a cell. But of course, the cellular machinery that these organisms evolved with was more complex and allowed them to deal with a wider range of challenges and opportunities in their environment. So just some basics here. So changes in temperature, changes in chemical environment, so radiation, toxins, other organisms that are competing with them, organisms that might be trying to eat them, resources they may not have. So we've seen examples of, for example, digesting different types of sugar might be required if you are an organism, find yourself in different kinds of food sources. Being able to make use of those is important. So basically, I think these entry slides, I want to give you the idea that what we term, what we call essential is, it's a little bit up for debate, right? So absolutely essential would be genetic information that you just can't do without in a perfect lab environment. Other genes are maybe a little bit more questionable how essential they are. Most of them, if not all of the functioning genes of an organism, are required in some context. So they've been useful, they stick around over evolutionary time, but not necessarily strictly required as these might be. All right, so I mentioned the numbers, relative numbers of genes that are required or essential. Think about here, what are the essential functions? So if you, again, like I said, strip down a cell to its bare minimum, all that it needs just to be a living cell, you get down to the very basic functions. So metabolism, being able to take an energy source and process that, being able to build the molecules essential for life, being able to put a cell together. So membranes, cytoskeleton, And then to do this process of central dogma. So if you have genes, you need to have the machinery to transcribe and translate them, to build the DNA and RNA and proteins and so on. So these are essential really in all cells, and they're functions that in a lot of cases are very common in between cells. So we have this evolutionary theory that says that life came from life. So everything from the original cell basically has had the same toolkit as a starting point and built on that as time goes on. So here's an example of some of the central dogma required DNA would be the genes that make up, as shown here, the ribosomal protein. So the ribosome is involved in translation, as we've seen, and those ribosomal proteins, if they are mutated too far, then translation itself doesn't work. And so that's a really major essential part of any cell, is being able to translate its RNA into protein. What shows here are the amount of genetic information that's shared among these different organism types with respect to their ribosomal proteins. There are lots of different ribosomal proteins, and groups down here, these domains of life, the eukaryotes, the archaeans, bacteria, and then mito is mitochondria in eukaryotes, so sort of related to bacteria ancestrally, but now living inside of eukaryotic cells. Shows sort of, you can see a block here, the split between archaea and bacteria gives a different color, different kind of ribosomal proteins here that are then shared and similar between bacteria and mitochondria. So it's sort of a basic core of very similar, and then everybody else mostly similar, and then a little bit of variation, of course, across organisms. So here's just an example. We compare these ribosomal proteins. Don't worry about the details, but I thought this was kind of cool from the study that's linked here. These like essential parts of ribosomal proteins and a variation around them. So the parts that are, as it's called, conserved core, these are little pieces of amino acid sequence that are the same across lots of different life. So bacteria, mitochondria, archaea, eukaryotes, and the parts in blue and yellow, those are areas that are different, sometimes very different in between these groups. So the function is conserved. The important parts of the protein are conserved, but other parts are able to change over time. So we talked about the number of genes that are essential in humans, again, under those strict cell culture conditions. Just a kind of a two ends of the complexity spectrum in eukaryotes. We've got these single-celled yeast that are eukaryotic fungi, and we've got multicellular humans. And people go in and investigate what are the essential genes and what do those genes do. So the chart on the right here, this is actually from a roundworm, C. elegans, and describes the functions of those. And just like we saw before, lots of central dogma. So protein synthesis, RNA synthesis, DNA synthesis, also basics of being a cell, cell architecture, metabolism, and it's down here. These are genes that are, they're shaded in black bars to indicate that they're more likely, significantly more likely to be important for life. If you remove them, then the cells die in these categories versus some of these other categories, which are maybe less strictly essential to a cell. And over here, I'm posing a question, why is there such a difference? So 1 in 5 yeast genes is essential, and closer to 1 out of 10 of the human genes are essential. So what is it about the difference in these organisms? You could probably guess that one. So yeast, just being less complex organisms, would have fewer genes that are maybe specific to different cell types. They're only one type of cell, specific to development or cell-to-cell communication, things like that. Humans are more complex organisms. I think that's pretty safe to say. And by having more complexity, they have more genes, and those genes are not essential for cell survival. They might be essential for whole organism survival, but fewer of the fewer of the genes, respectively, by a proportion, are actually required for the life of one cell. So lots of extra genes out there in the humans. Another perspective, a little bit relating to what we saw with the ribosomal protein genes, are that we have some similarity across groups in proteins that they make and the functions of those proteins. And there's a term on here, orthologs, you might run into once in a while. Genes that are orthologous, those basically have the same function because they inherited that function from a common ancestor. So here's an example of proteins used to wrap up DNA in the nucleosomes and histones of what we have in our cells. And then this is a very distantly related organism in archaea, has something kind of similar, kind of related. And this paper was hypothesizing those are orthologous genes. So I mentioned a few times, I think it's worth throwing it on here, the domains of life. I often ask students if they've been briefed on this, and a lot of people give me a blank stare when I ask them about the domains of life. So when I was going through school, we had the kingdoms of life. And these days, the most basic split in living organisms is in these categories called domains. And seems like nothing lasts forever. There were 3 nice and clean domains for a while there, and this middle domain has gotten a little bit fuzzier, but we have definitely this group of bacteria. So this is a domain of life known as bacteria. Up here we have a domain of life known as eukaryota or eukarya. And then there's a bunch of in-between stuff that doesn't have a single branching point necessarily. And these are the archaea. So they've gotten a lot more attention these days, which is great. They're organisms, a lot of times in the far-flung regions of the world, thermal vents, things like that, sometimes have some of our archaea. And they're very different. They're evolutionarily, as far away from humans as bacteria are. And they're still kind of piecing together how they're all related to each other. So I think for now we can probably stick with the 3 domains of life. Just kind of keep in mind that it's getting a little more complicated. And I throw this on here. There are, I believe they're kind of new categories. And they've come up with these really important big divisions of life within the archaeans. And they had to give them names, and they've come up with some fun names based on mythology. So over here in red, we have the Asgard archaea. So this is somebody's cartoon drawing of Loki archaeota. So they've named them after some characters you might recognize from your superhero films. We've got Thor archaeota, Loki archaeota, Heimdall archaeota. And some of these are actually from, there's Odin. They're coming in from other mythologies of other cultures around the world. So just totally sidestep topic here. Anyway, lots of diversity in the archaeans. Some of that relates in interesting ways to our cells because basically the original eukaryote cell was kind of an offshoot of archaea. So it's useful for learning about our origins to study a little bit more about the living archaea. So kind of in that relationship, this is a set of genes with a common ancestor, and the original genetic information was for making a tubulin protein. So tubulin is part of the cytoskeleton, and very important, also very ancient, right? So the first cells had some first cytoskeleton, and in the eukaryotes, there are these different colors here, the alpha tubulins, beta tubulins, gamma, delta, zeta, eta tubulins. Big diversity of those among the eukaryotes, and then interspersed with those are also some variation showing different bacterial versions. And over here we've got some archaean tubulins. So when we look at essential genes, a lot of times the essential genes are the ones that have always been essential, going back to the first cells. So some original cell came up with the idea, or came up with the ability to have a cytoskeleton that was important and essential, and everything that descended from that original cell also had a cytoskeleton. So it may be diversified in the ways it can make cytoskeleton proteins, but still has this version of ancestral cytoskeleton there. So that's one of the trends we see is diversification over time. So these in here are some varieties of eukaryotic tubulins, again with these Greek letters. And maybe you recognize there's some scientific names abbreviated here. So darn, I had it. Here we go. Dromet, that's Drosophila melanogaster. So that's our fruit fly in there. Musculus, the mouse. Humans are in there somewhere, maybe. Anyway, lots going on there. So kind of working toward ourselves in the global phylogeny of life, getting closer to humans, there are some specific genes that are specific to humans. So we have lots in common with our cellular distant relatives, bacteria, archaea, because we've got some carryovers from those original cellular components. And then as organisms diversify, they get new abilities, new genes, and we've got our share of those. And this is from this headline here, mentions how a lot of the human-specific genes are specific to functions that we think of as uniquely human, or certainly characteristic of humans relative to our close relatives. So brain, immune, and metabolic function. So humans are definitely, we stand apart from a lot of animals in having brain function, and it's like superior brain function, we like to think. And sure enough, there's genetic evidence for the origin of these human brain functions in the original humans. This is from another study that talks about basically just as the branching happened, this is highlighting the mammals, as the mammal tree branched out, this is showing along the branches how many new genes came into existence along those branches. So we have features that we can say are distinct of the various lineages of animals, and having these distinct features also tracks to having the genes for putting those together. So you can kind of trace your way up to humans. We've got here the monotreme in the base, the platypus, big split, 172 genes on our way to the placental mammals. Here's marsupials over here, 36 distinct marsupial features. The other, forget which ones are placental, I'll be honest. The other mammals. And then working our way up to, buried in here, Homo sapiens there. So the branch leading to Homo sapiens, not too many distinct, 49 distinct genes according to this study, heading off to Homo sapiens. So the difference is, of course, as we know, organisms are different from each other. They may have different versions of genes, and they also have different genes that maybe didn't exist before. And this is a study that shows basically how a new gene might come about. So again, genes aren't just like lightning into existence. They come from existing genetic information. And the easiest way to make a newly functional gene is to take pieces of existing genes and put them together. Sometimes genes are copied, and those copies become functional new versions. And this is from an interesting piece of evidence where they found one of these distinctive human nerve function, brain function genes actually related to something that still exists in our ape relatives, but doesn't work in the same way in those relatives, the chimpanzee and the gorilla, and does work just fine in the human. So kind of a dramatic title here, rescued from the DNA garbage bin, a genetic feature that allows humans to have our big brains. Actually relates to just a little difference in the coding sequence of this gene, N2NLR. So we talked about essential genes, And some of the, it's kind of a mid-range of not strictly essential for life, but mutations in these genes are known to influence basically health and survival. So I got a list of these from the internet of different gene regions that are single genes that are linked to some of our major genetic disorders. So lots of human health has to do with environmental conditions and exposures. And then a decent dose of human health concerns are inherited. So knowing the causes for those can help us address the potential cures for them. Previously in the mutation lecture, we talked about cystic fibrosis. And so basically these are genes that, if mutated, can cause deficiencies that may jeopardize the health of the organism. So a lot of times there's a lower average life expectancy for people who have these diseases. And so kind of looking at that from this last bullet point here, from the perspective of evolution, these are differences that these days are maybe to a little bigger extent manageable with medication and treatments and societal benefits that we have these days. But say in a pre-civilization human, these might not have been something that would have allowed someone to survive and pass on the genes. So we're in kind of an interesting place right now where we have a great understanding about how our DNA works. Also has opened up the ability to keep some of these harmful mutations around because we can allow someone to live with those and potentially even to pass them on to their children. So for example, for survival and reproduction, as long as a person can survive to an age of reproduction and have children with these genetic components, then that would be something that they can pass on to their kids. Some of these, quite a few of these, can exist as recessive alleles, so they may exist in the population and not cause disease to the people who have them, but they can again still pass them on to their children. So these are kind of in the middle of essential, non-essential, certainly a major impact on quality of life for somebody who has some of these genetic conditions. A little bit more, maybe heading more towards minor effects. So I mentioned basically the influence on survival and reproduction. These are some things that we've shown before. So lactose intolerance, probably key to survival, as I've mentioned, in cultures that are, if you're raising dairy cows and your civilization is dependent on nutrition from milk and cheese to get your nutrition. If you have a distaste for milk and dairy products, then that might set you back as far as getting your proper nutrition. That could be detrimental. So maybe selection that favors lactose tolerance later in life. Distaste for cilantro, not so sure about that one. Maybe you could certainly construct a reason why that could be adaptive or maladaptive. So not being able to eat, I don't know, one of your plant options around you could be a problem. Other ones, pigmentation, so skin and eye pigmentation, we've mentioned a few times, has been linked to sun exposure, so increased incidence of skin cancer in individuals with lighter skin. And then on the flip side of that, people with lighter skin have better ability in low light conditions, like far northern latitudes, have a better ability to turn sunlight into vitamin D because more of that light is getting into their skin. So it's sort of a two-part selection there on skin color and eye color, and we see that in the ancestral human cultures from different parts of the world. Mentioned a while back, I think our first time, we talked about this potential gene that causes people to cheat on their significant other. You could, I don't know, maybe construct a scenario where that would be more adaptive or maladaptive, again, as far as getting your genes around, maybe. Differences in height, exceptionally tall, exceptionally short, maybe affects on survival in the long term. So kind of middle range, genes of middle range effects, and then kind of all the way down, there are pieces of DNA throughout all kinds of organisms that we don't really know what they do. And sometimes they get this interesting name of ORF, which is open reading frame. We have a lot of DNA sequence for genomes, and within that you can see pieces of DNA that look like a gene. They might have a piece that looks like a promoter and a long stretch of DNA that could be coded, translated into a protein, but we don't actually know what the region does. And so for a lot of these, they're kind of flagged as potential genes. And I just happened to grab one. This one I thought was interesting. It's called open reading frame, comma, dubious. So good chance of not actually having a function. So there might be, you kind of see this pretty small element there. This potentially could make a protein, but maybe that protein doesn't do anything, or maybe it never makes a protein. And that's a current pursuit. So we have these big genome level DNA sequence databases, and we don't know what every piece of DNA does. And people are actively investigating, just as we've seen before, what happens if I delete this? Can I delete it under some context and find out that it was actually important to that organism? So here's a yeast example. Here's another yeast example, heading through time and kind of tapering off with the discovery of new functions for these proteins. So we have 100% possible of proteins that we don't know what they do, and the number that we don't know what they do is trimming down, kind of leveling off at about, I don't know, 12% of proteins. We don't quite know what they do. So either they're not turned into proteins maybe, or they just have a function that's so minor that we can't really detect it with our tools of looking at things genetically. So almost there, finding out what everything does. Still lots of room to investigate.